Hello friends, welcome back in the channel of Clinical Care Insight. We started a series of lecture on Introduction to Microbiology. Until now, we discussed about what is the meaning of microbiology. In the microbiology, what things we are going to study, okay? And we also seen about the major branches as well as the scope of microbiology. So in today's lectures we are going to discuss about the historical aspect of microbiology, and here is today's lecture outline. First we will deal with the introduction, then we will define the microbiology history under the the time period. The first one is the discovery era, okay, another one is called transition period or transition era, and the next one is the golden age of microbiology, okay. And after the break we will discuss about the microscopy. Basically we will define different types of microscope, okay, and their uses. So start today's lecture with, make the heading historical aspect of microbiology. Historical perspective. One thing you should clear in your mind, since the microbes are very small, isn't it? It's a very small, and we already know that we cannot see the microbes by our naked eye. Okay, so there's a need for the proper, the magnifying apparatus, for need for, the one standard microscope in order to see the microbes. Okay, so initially the microbes are invisible. Okay, microbes are invisible, and traditional persons, they are thinking, okay, the diseases which is the present in the humans as well as the animals, they are the divine creation. This disease happened due to the divine mercy. Okay, so that's called the religious dogma about the disease. Okay, but time to time, that time is ever dynamic, okay, and also the science is a dynamic field, dynamic discipline, okay, and there is the constant research, okay, constant discoveries in the field of science, okay. So what happened initially, the microbiologists or different researchers, like some of come from the physics field, some of comes from the chemical, the chemistry field, okay, and they contributed their effort to understand, to establish one amazing branch, and that branch is called as the microbiology. That branch is called as the microbiology, which deals with the studying of the microbes. So for understanding the historical aspect or historical perspective of microbiology, we will categorize their history into the three time period. So the first time period you should write it down, is called discovery era. First one is called as the discovery era. Era means period, okay. Another one is called as the transition era or transition period. Transition period, or you can also write the transition era, okay. And the last one is written down, referred as a golden age of microbiology. Golden age of microbiology. So first I will tell you the superficially, then I'm going in detail to describing these periods, okay, describing this period. The first thing you should keep in your mind that during the, starting from the 15th century, 1567 or 1558, scientists were, or different field persons were involved in understanding, or discovering the apparatus, the magnifying apparatus, in order to see the things, not the microbes, because they had in their mind that the microbes are you can say they are not a causative agent of, they are not a causative agent of the diseases, okay, and they are the divine's mercy. The diseases are for the divine's mercy. So initially the discoveries of the, you can say, magnifying apparatus, they are dependent on seeing the other objects, they are dependent on seeing the different, you can say, the object. So the discovery of the first microscope is, you can say, contributed by one physicist, okay, by one physicist, okay, called as the Galileo Galilei, fine. And then there are periods came where there is a, you can say, a huge kind of misconception that the life forms which is, you can say, existing in this planet, they comes from the non-living objects, they come from the non-living objects, and that is called as the abiogenesis, that is called as the abiogenesis. And now this abiogenesis, see, they are saying that the life forms which is present in this planet, they comes from the non-living object. That supported the religion's theory that the diseases is not caused by the microbes, they are, you can say, divine, you can say, creation, or it's a fault of a person who is suffering, that's their fault, okay. God is, you can say, punishing him or her by, you can say, making them ill. Are you getting the point? Yeah. But once the, you can say, the microscopes are started to develop, okay, there is a clear-cut idea that there is something called as the microbes which is existing in the you can say, in the across the planet, and they are responsible for causing the disease, okay. And these linings come from the one theory called as the germ theory of diseases, called as the germ theory of diseases. But eventually in this period only the microscopes are discovered, and in transition period there will be something called the abiogenesis, okay, or theory of spontaneous generation, which claims that the lives come from the non-living object. And in this period that then the germ theory is, you can say, not able to establish properly, they are in a controversy, but during the golden age of the, you can say, the microbiology, what happened, some of the microbiologists, they are involved in establishing the clear-cut microbiological principles, okay, which prove that the diseases are caused by the microbes, or there is an absolute existence of the microbes, okay, or there is a proper involvement of the microbes in the case of the diseases, okay, in case of the causing the diseases, and those category of microbes are referred as the pathogens. So let's make the first heading discovery era and understand what things is discovered in this time period. So as I said, the discovery era is devoted to, you can say, discovering the microscope, fine. So the first microscope is discovered by the Galileo Galilei. The first microscope is discovered by the Galileo Galilei. He is a known physicist, okay, he is a known physicist, and he discovered the, you can say, the first microscope, okay, no problem, yeah, it's a Galileo Galilei. Now there was the another, you can say, a person, okay, who discovered the another, you can say, high magnifying, comparatively high magnifying microscope, and the person name is, yeah, very good, Antony von Leeuwenhoek. Antony von Leeuwenhoek, okay. Actually he was a tailor, he was a cloth, you can say, a cloth tailor, and in order to see the fibers, the cloth fibers, he discovered the microscope, and that microscope is called as the light microscope, okay, light microscope. And this light microscope, they are also called as the optical microscope, They are also called as the optical microscope. Getting the points? Yeah. So it is the Antony von Leeuwenhoek. It is Antony von Leeuwenhoek, and it is the Antony von Leeuwenhoek who discovered the first microbial life. That there is a existence of microbes. Understood? They studied about many bacteria, yeast, and protozoa. Okay, and by this their contribution, they are referred as the father of microscopy. Okay, they are called as the father of microscopy. You should put on a star mark and write it down. He is called as the father of microscopy. In the competitive exams, you can say they were asking that who is the father of microscopy, who discovered the light microscope. Understood? And even like examiners asking about their profession, which fields they come from. Either they are a physics, tailor. Getting the points? Yeah. So you have to understand this. You can say that right this you can say the terms that he is a father of microscopy. He is a father of microscopy. But there is a problem. The problem is the light microscope can see the microbes. Okay, by this microscope we can see they providing the magnification. They providing the magnification ranging from the 10x to 1000x. 1000x, that's their magnifying, you can say, the range. But this light microscope, they are filled when the study is about the internal structure of the cell, about the internal structure of the microbes. So for that there was another microbiologist called as the Ernst Ruska. Okay, known as the Ernst Ruska. Ernst Ruska, okay, and he discovered the electron microscope. So the first microscope is discovered by the Ernst Ruska. Okay, and the difference between the electron microscope and the light microscope is that the electron microscope generate the 3D images. You can visualize the organism in the 3 dimension. The first thing. Second thing is that through the electron beam, you can see the, you can say, microbial structure or human cell structure in a clear-cut way. Okay, very precise way. Got the point? And there are types of electron microscope. There are 2 principal types called as the TEM and SEM. TEM refers to the transitional electron microscope. Okay, transitional electron microscope, and another class is called as the SEM. Scanning electron microscope. These things we will discuss in detail in the microscopy, but for a while you just understand that the first period, if we talked about the history of microbiology, the first period is called as the discovery era. Here what happened, the scientists, you can say, or different fields persons, they are actively involved in discovering the proper magnifying apparatus. The first person is the Galileo Galilei, who is a profession-wise physicist. Okay, he discovered the first microscope. He discovered the first optical microscope. Now comes to the another one. Another one is the Antony von Leeuwenhoek, A.V. Leeuwenhoek. He is the one who were seen the first live cell. Okay, and profession-wise he is a tailor. Okay, and he discovered the first light microscope, also called as the optical microscope. You can say the property of the light microscope is you can magnify the object ranging from 10x to 1000x. Got the points? But you can say this microscope, they failed when it's come to study the internal structure of the cell. So for that there was another, you can say, the scientist, that is his name is the Ernst Ruska. His name is the Ernst Ruska, and he discovered the electron microscope. Electron microscope is better than light microscope just because electron microscope uses the electron beam and generate the 3-dimensional view of the object. Fine, no problem. Now, once we understood about the discovery era, let's switch on and study the transition period.